So what I do in a review is I read the question, and then you tell me what you think is in the blanks or how to answer the question. And you may have to, I hear some feedback from somebody, you may have to turn down your volume or something a little bit, or wear earbuds. But anyway, as we go through this, you tell me the answers, and then I'll talk about it or answer any questions that you may have. Okay, any questions to begin with? Okay, so number one, it says blank complements blank. So what did you get for the first blank? Blank complements blank.
Structure complements function.
Okay, so you could say structure complements function. We could also say anatomy complements physiology, since anatomy is structure and physiology is to study the function of how our body works. So we could use either of those in the blank. And then it says this blank between form and function is an important theme to keep in mind through our study. So the idea here is, what do you think the second blank was?
Relationship.
Yeah, it's relationship. It's the relationship between these. And so what two questions, this is just to expound on it a little bit. What two questions are we always trying to answer as we go through the course? Well, what we're trying to do is we're trying to ask ourselves two basic questions. What is it and what does it do? That's what we're always asking ourselves. What is it and what does it do? And that's anatomy and physiology. Okay, number two, arrange the levels of human structural complexity from the smallest to the largest structural units according to the levels listed in your notes. So what would those levels be? What's the very basic level that all things, no matter alive or not alive, non-living, what are all things made up of?
Atoms.
Right, atoms. And so atoms bond together through chemical reactions and form molecules. And then we have two different sizes of molecules. So I talked about that in the lecture. We have small molecules called
Macromolecules.
Okay, small molecules are called monomers. Okay, and then large molecules are called polymers. And yes, they are macromolecules. So large molecules are polymers or macromolecules. Okay, and then these macromolecules, there's four categories or classifications of these. And so we have carbohydrates, lipids, proteins, and nucleic acids. And inside of cells, they make up the organelles. So the next level is organelles. So we've gone from we've gone from atoms to molecules to organelles, and now we have cells. And what's unique about cells? What's special about them? Cells are
Living,
right, and everything below the cellular level is non-living. So you're actually made out of non-living material, which is amazing. That's the miracle of this whole biology, is that we're alive and we know it. Then cells bond together and they form different tissue layers. Tissues are cells that are clones of one another. They're all identical in the way they appear, and they all do the same task. And so in lab, you just had your lab test, you had lots of tissues on that lab test, and some of you did real well on it, and some of you struggled with that. Tissues is kind of your first complicated study. It takes a lot of study to go through that, but there's entire courses on tissues called histology. If you went to a four-year university, you could take histology and just study all the tissues of the body. There's really hundreds, you only learned a few. And then tissues, different kinds of tissues make up organs. And so you could think about the heart, for instance. You haven't got to that yet, that's an AMP2, but the inside of your heart is lined with simple squamous epithelium. That's a tissue you learned. And then the bulk of the heart is made up of cardiac muscle tissue. And then there's lots of connective tissue, fibrous connective tissue that's around the heart that holds it together and on the outside of it. So multiple tissues make up organs. And then organs have classified in the past into systems. So we have organ systems. And does anybody remember what I told you in the notes about organ systems?
They don't actually exist except for educational purposes.
Exactly. So there's really no such thing as a system. Systems are just textbook chapters. That's all they are. Really, your body is one body, but we have systems so that we have sort of a systematic way. See, a systematic way to break down the human body into neat little chunks so that you can talk about it. And of course, your whole goal, if you're going into the nursing program, if that's what you're going to do, your whole goal is to eventually see the body as one body, so that if you're asked a question about what happens if someone's carbon dioxide levels rises too high in their bloodstream, you would be able to name every system of the body that it would affect and how their physiology would change, and then in nursing how to correct it. So you're trying to see the body as one unit, one whole unit. Okay, then after organ system we have organism. Okay, so organism at the top. Okay, moving on to number 3. So if you got your review, number 3, the smallest structures that biologists consider being alive are
Cells.
cells, right? Cells. Number 4, a or an blank by definition is composed of 2 or more types of tissues.
Organ.
Organ.
Organs, right? Organ. Number 5, the process of chemically changing nutrients and incorporating the resulting molecules into the body's tissue is what?
Chemical digestion.
Oh. Anybody have a different?
No.
No? Okay, so chemically changing nutrients, and the key word here is incorporating the resulting molecules into the body. So digestion is just a process of breaking down. So what do we call taking nutrients and making them part of us? It starts with an A. It starts with an A.
Is it assimilation?
Yeah, it's assimilation. It's the life process of assimilation. So in simple terms, assimilation is you are what you eat. Yeah, maybe you had a mother or grandmother who told you that you are what you eat. Hopefully, if they were worried about your nutrition. And so if all you ate was junk food, then that's what your body's made out of, because what you eat assimilates into your body tissues. It becomes you. It becomes you. And it usually takes about 3 months. If you change your diet to try to eat healthier, it usually takes 3 months for you to see a result in your body and muscle and skin and things like that as your body changes and becomes new material. And it's kind of interesting. It's fascinating to me that your body, you realize your body is constantly repairing itself and cells are being replaced. With many, about 80% of the cells, many of them are being replaced every 3 months. So all of you right now have a brand new body. You have a brand new body. Your body, you know, however old you are, if you're 28 years old, if you're 19 years old, if you're 30 years old, your body's really only about, most of it's really only about 3 months old, which is amazing. You're constantly being replaced. It's just every time you're put back together a little bit more disordered, and it gives you the appearance of aging. That's fascinating to me. But anyway, you're not really aging. You just appear to be, and you're put back together more disordered. But your atoms are new to you. They're being replaced. Okay, let's see. So the assimilation. So that's a long explanation, but 6, the process by which organisms release substances such as vitamins, hormones, and saliva. What is that?
Secretion.
Secretion.
Secretion, right? Secretion. And then define ingestion. So what is ingestion?
It's food getting, is how you put
Yeah.
it in the notes.
Right. This food getting, it's the idea of nutrients, that you're acquiring nutrients. And every living thing has to acquire nutrients in some way. Okay, 8, the process by which waste products are eliminated from the cell by diffusion through the cell membrane is called?
Excretion.
Right, excretion. So excretion is the ability to get rid of waste. Don't get mixed up with secretion. Secretion and excretion sound a lot. Secretion is producing useful things like vitamins and hormones and saliva, tears, and digestive enzymes in the duodenum. That's secretion. But excretion would be getting rid of things that are potentially harmful. And here's one thing that you can remember. What is harmful to your body? Well, some people would say, well, waste. But in reality, what is harmful is anything out of homeostatic range. Anything that you have too much of is harmful. What's the difference from a poison and a useful drug? What's the difference from a poison and a useful drug? You know? The difference is the dosage. That's the only difference. As a nurse, if you're going into nursing, most of you are going into nursing. If you're going into nursing, realize you're going to be actually giving people poisons all the time. All drugs are poisons. But you got to get on dosage. So you give them just the right amount. Otherwise, you're poisoning them. Okay, so it's true for food too. You realize all food that you eat can potentially be life-threatening if you eat too much, just like water. Water can be deadly if you take too much. Everything has to be within homeostatic range. Okay, 9 says define egestion. So what is egestion?
The void of undigested materials.
Yeah, so we eat things all the time. What is one thing that we eat all the, or it should be if you're eating healthy, that you eat that you egest?
Fiber.
Fiber, right. So we cannot digest the cell walls of plants, and so it is egested from our body, but it's useful when that happens because it keeps our feces from being like a brick when we pass it, which would not be very pleasant. 10. The process by which cells chemically break down small food molecules and release chemical energy for the cell's use is?
Digestion.
Okay, so the key in 10, anybody got something different?
Respiration.
Respiration, that's right, respiration. The key in number 10 is chemical energy. It's the idea of releasing chemical energy, and all organisms must have some source of energy, and that is a process of respiration. And through cellular respiration, we produce a molecule that we call, starts with an A?
ATP?
ATP.
Yeah, ATP, right, ATP. It's an acronym, adenosine triphosphate. Okay, 11, number 11 is a list of applied sciences and the medical fields. Does anybody have a question about those? I think they're pretty basic. They're defined in your notes.
No, sir.
So, okay, so it should be pretty plain to look those up. The whole idea of going over those is to give you an idea, if you're going into the medical field, maybe of which direction you would like to go to know the medical fields. Okay, number 12, it says the blank states that energy cannot be, well, I just told you. The blank states that energy cannot be created or destroyed. I almost told you. What is in the blank?
Law of conversation.
The law of conservation.
Yeah, the law of conservation of energy. It's called the law of conservation of energy. There's also a law of conservation of mass, but this is the law of conservation of energy. Okay, 13. So getting now more into atoms and the chemistry side of things. 13, the atomic number of an element indicates the number of?
Proton.
Protons in the nucleus.
Yeah, that's right, protons. Protons of a specific element. So does the atomic number indicate electrons?
Yes.
Not always.
Not always, that's right, not always. It definitely indicates the number of protons, but not always electrons, because electrons can be lost or gained, right? Lost or gained. Okay, 14, what condition must exist for an atom to be electrically balanced?
Equal number of protons and electrons.
Valence shell must be complete.
Okay, so to be chemical, so we had two different answers. One answer is protons and electrons must be equal, and the other answer is the valence shell must be complete. So which one is the right answer? Both of you are referring to things that are balanced. But to be electrically balanced, we have to have the same number of protons and electrons. Electrical charge, think about positive and negative electrical charges like on a battery. So we're talking about protons which are positive and electrons which are negatively charged, and they must be balanced in equal number. To be chemically balanced, there must be a complete set of electrons in the valence shell. Okay, so there are two different things, electrically balanced versus chemically balanced. Does that make sense? Anybody have a question on that? Okay, so the answer for 14 is an equal number of protons and electrons. Okay, 15, what are ions?
Charged atoms.
Charged atoms. How are they charged? What made them charged?
They lose or gain electrons.
Right, they've lost or gained electrons. That's what makes them. So ions, does anybody know the synonyms for ions? Ions are also called?
Cations or anions.
Okay, now that's specific types of ions, either positively charged or negatively charged. Cations are positive and anions are negative. But what I'm thinking of is you need to know the synonyms of an ion. So an ion is a charged atom. If you're going into the medical field, you're not going to call them ions. Like if you're in a hospital environment, or if you're going to nutrition, you wouldn't say, make sure that you get all your ions. What are you probably going to call them instead of ions?
Electrolyte.
That's right. So we could call them electrolytes. That would be one,
Salt.
electrolytes. Salts, right? And what's another thing that probably everybody calls them and not realize what they're talking about? Starts with
Is
an
it
M.
carbohydrate? Oh.
No, it starts with an M.
Mineral.
Minerals, right, minerals, your daily minerals. Vitamins and minerals. Minerals are ions. Now, vitamins are actually biological molecules, something entirely different, but minerals are ions. So electrolyte, salts, minerals, ions. In an IV bag, there will be minerals or electrolytes, which are ions. And you're going to have sodium and potassium. Sodium and potassium are going to be two basic ones, along with calcium is going to be present. But you'll really need to understand your ions if you're going into nursing. Okay, 16. Number 16, how can radioactive isotopes be used in the medical sciences?
As
For
tracers.
PET scans.
Yeah, so a PET scan as tracers. If you've seen images of people being injected with different kinds of dye, sometimes those dyes are radioactive isotopes. Sometimes different medical treatment we can use radioactive isotopes. Certain kinds of cancer treatments use radioactive isotopes. 17, what are valence electrons?
Number of
Outer
electrons.
shell electron.
Okay, so it's the number of electrons in the outermost shell of an atom. And so I give you a scenario here on 17. If an atomic number of potassium is 15, it says how many electrons are found in the valence shell of this atom in its non-ionic state. Okay, so what non-ionic means is that it has not lost or gained electrons. It's just as it appears on the periodic table.
Would it be
So-
5?
5.
That's right, 5. Okay, so we would have 2 in the first shell, 8 in the second shell, and 5 in the third shell. Okay, so what if I ask you this question? If an atom had a total of 15 electrons, what if I said how many electrons are found in its valence shell in its ionic state?
8.
And the answer is 8. That's right, it's 8. Now, what if I ask you this question? What if I said if an atom has 15 electrons, what is the charge of the atom in its ionic state?
Negative.
Positive.
Okay, it's negative what? So remember, there's 2 in the first shell, 8 in the second shell, 5 in the third shell. the octet rule says we have to have a total of 8 in the outermost shell. So is it easier to lose 5 or to gain 3?
Gain 3. So
Gain
it'd be
3. So
-3.
-3, that's right. So the atom would have a -3 charge in its ionic state. Okay, so if you didn't catch that, I drew out atoms in my videos and went over that. You might want to go back and watch some of that, or watch some of the tutorial videos that I put out. Okay, 18. The blank states that except for the first energy shell, atoms are stable with blank electrons in their valence shell. So it says what's the first blank?
Rule of 8.
Octet rule.
Okay, the rule of 8 or the octet rule, that's right, it can be called either one, states that except for the first energy shell, atoms are stable with
8.
8 electrons. 8 electrons. So 2 in the first shell and 8 in all the others. It's stable. Let's see. And so A says the numbers listed represent the first, second, and third energy level respectively. On this basis, which of the following 3 is an unstable atom?
The second one.
The second one. That's right, the second one. Good. B says atom X is electrically balanced and has 10 protons. How many electrons would it have in its valence shell?
8.
8.
And so the answer would be 8. That's right. So 2 in the first shell, 8 in the second. It's electrically balanced, meaning it has the same number of protons as electrons. And so 2 in the first shell, 8 in the second. C says atom Z has an atomic number of 74. What can you determine from this number?
The number of protons.
That's right, it has 74 protons. 74 protons. And then D is a practice. It says be able to draw planetary models of atoms of elements 1 through 18. So this is hydrogen through argon. Some of those are shown in your textbook. I just give you some symbols to use for practice, and if you do that, you'll be going along with the video that I put out for you to watch. Anybody have questions on that?
Are we going to be able to use a periodic table on the test?
So on the test, in the past, what I've had, of course, if it was a face-to-face class, I've had you draw. I would have you draw maybe a couple examples, but since this is online, I will give you anything you need to know, and you won't have a periodic table because I'll give it to you in the problem.
Okay.
What you need to know. So, but in the past, I've passed out a periodic table with the test. 21, the formula H2O means that there are blank hydrogen and blank oxygen atoms.
2 hydrogen.
2 hydrogen and 1
That's
oxygen.
right, 2 hydrogen and 1 oxygen atom. Just 22 now. Chemical energy is an example of blank, which is stored or inactive energy.
Potential energy?
Potential, that's right, potential energy. 23, a blank is an atom that has lost 1 or more of its electrons.
Cation?
A cation, that's right, cations are positive, and if an atom loses an electron, it becomes positive. Not because it gained a proton, it's just because it now has more positive charges than it does negative when it loses an electron. So sometimes that sounds kind of weird to say that when an atom gains an electron, it becomes negative, and it loses an electron, it becomes positive. That sounds strange, but if you understand it's all about charges, and it's not about math, you know, when we're thinking about charge, then that makes sense. Anybody have questions?
No, sir.
Okay, let's see. Next question is 24. Salts like sodium, sodium chloride, table salt, or hydrogen sulfate or magnesium sulfate are.
Inorganic.
So these are. Anybody know what kind of compounds they are?
Inorganic?
Inorganic?
So maybe somebody said inorganic. So inorganic compounds. They contain, they're also ionic compounds. So either one of those could go in the blank, inorganic or ionic compounds. '25, the chemical behavior of an atom is determined by the blank in its outermost energy shell.
Number of electrons.
Yeah, it's electrons in the outermost shell. So it's all about the only electrons that play a part in bonding is the outer electrons in the valence shell. So the chemical behavior of an atom all depends on the outermost shell. Okay, 26. In an blank bond, the atoms completely transfer electrons from one to another.
Ionic.
Ionic.
Ionic, right. So an ionic compound. And then the next one says in a blank bond, the atoms share electrons.
Covalent.
And so that is covalent. And so these are very different bonds, and I tried to explain this in the video why they're important. Because in an ionic bond, once atoms transfer electrons from one to another, they don't have to stay together anymore. They're stable and they can separate from one another. But covalent bonds can never separate. The atoms that are covalently bonded together can never separate without some sort of enzymatic action. They are permanently partnered together because they must share. If they try to separate, they would become unstable and they would just snap back together. Okay, 28. A blank bond is a weak chemical bond formed when a partially positive hydrogen atom in a molecule is attracted to a partially negative atom in another molecule.
Hydrogen.
Hydrogen, that's right, a hydrogen bond. And it is most easily seen between what kind of molecules?
Water.
Water.
That's right, water molecules. Water molecules. Okay, next on 29, the next page. What do the chemicals, chemical symbols, and so this is H-H. So there's a little line, one single line. So H-H and O, and then two lines, O, and then N and 3 lines and N mean. So
Means they're
what are,
sharing electrons.
that's right, they're sharing electrons. Those dashed lines means the sharing of an electron, and every dashed line represents one electron. So when 2 hydrogens share an electron, we use a single line. That's called a single bond, a single covalent bond. When oxygen shares electrons, we have 2 between them and an oxygen molecule, which would be written out O2, an oxygen molecule. So you're going to see O2 quite a bit in different places in the book. So it would be O2. So you're going to see that written out. And then N, which is nitrogen, when it shares a bond with another nitrogen atom, it forms a triple bond, which nitrogen gas is into, and nitrogen gas is the most abundant gas in our atmosphere. So that's going to be important coming up in AMP2, these gases, when you talk about them. But make sure you realize that we're dealing with bonding here. This is bonding. It's not, I remember asking this on a test question where I have an O and then I have 2 dashed lines and an O, and I ask, what was this? And some student said it means 0 equals 0. Well, that's not what it means. Okay, so make sure you know that these are the sharing of electrons. Okay, 30. What is the difference from an endergonic and an exergonic chemical reaction?
One
Endergonic
absorbs heat.
absorbs heat and the exergonic releases heat.
Right, so ender means, it is Greek for inside, pulling in, and then you have this same root word ergo, which means work, so it's pulling in. It's during the chemical reaction, it pulls in energy, it pulls in the ability to do work. And then exergonic, it comes from the Greek ek, epsilon kappa, which means out, going out. So exit, that's where we get the word exit. So work is going out, energy is going out from it. Exergonic reaction. 31. What influences the speed or rate of chemical reaction?
Catalyst.
So the presence of a catalyst, that's right. What else?
Temperature,
Temperature,
Concentration.
and size.
concentration, right? Concentration of the reactants. That will influence the rate of reaction. Those are some of the main ones that I listed. 32, the reaction of A plus B yields AB is an example of what kind of reaction?
Synthesis.
That would be a synthesis. So a synthesis is also called a combination reaction. It's also called an anabolic reaction, which means to build. So all of those terms work. 33, the reaction AB plus CD yields AD plus CB is an example of
Displacement.
displacement or exchange reaction. Displacement or exchange. 34, monosaccharides joining together to make a disaccharide is a good example of a,
Synthesis.
so we could say a synthesis reaction. And what kind of synthesis is it, since we're dealing with organic molecules? Specifically, it's a type of what?
Dehydration.
Yeah, dehydration synthesis between the two. Okay, 35. It says two good examples of a colloid would be Jello or
Cytosol.
cytosol. That's right, cytosol, which is the fluid inside of a cell, a cytosol. So Jello is actually a matrix of protein. Do you know where Jello comes from? Anybody know where Jello comes from?
Gelatin.
Yeah, where does gelatin come from? Anybody know? So it comes from bone marrow. That's where it comes from. So after after a cow goes to a slaughterhouse and they take all the steaks off the cow and what's left over is the bones, they're going to process all the bones, they extract the marrow, and they produce gelatin from the bones. Most of it's from beef. And so this was gelatin. Some people that grosses them out. It's not really. Jello was invented by a doctor well over 100 years ago as a source of protein to feed patients who had yellow fever or cholera or smallpox or something like that, so that they could get a source of protein that was easily digestible. And so Jello is still used in hospitals for that purpose. Jello's not bad for you as long as you don't add a lot of sugar and things like that to it. But it's made up of all these strands of protein, that are going in different directions, and it stays suspended in a watery matrix. So if you were to dry Jello, if all the water comes out, it turns back into a powder again. So, but anyway, Jello. 36, tell whether the following solutions are considered an acid or a base. So pH of 9,
Base.
that would be a base. pH of 2, pH of 2,
Acid.
acid. What about 7?
Neutral.
Neutral.
Neutral. 8,
Base.
Base.
that would be a base. 12,
Base.
base. What about 7.4?
Base.
Base.
Yeah, it would be slightly basic or a weak base, And of course, that's the pH of our blood, right? 7.4. It must be maintained at 7.4 for all the proteins in our blood to work correctly. If that pH changes even to 7.35 or to 7.45, going up or down, we're either going to metabolic acidosis or alkalosis. And it can eventually lead to death if it's not brought back to a range of 7.4. 37. What are the differences between mixtures and compounds? Anybody know this one?
Compounds you can't take apart.
Well, you can chemically. You can take them apart chemically. So if you have to deal with them chemically, what's a mixture not?
Not a chemical change.
Right, it's not, it's just things combined physically. There's no chemical reaction between the substances combined, where compounds are substances that have been chemically combined. That is, their electrons are bonding, their valence electrons are reacting. Okay, 38. What will happen to the pH of water if magnesium hydroxide is dissolved in it? Will the pH rise or drop?
Rise.
It will rise because magnesium hydroxide is a base. So the pH will rise. If a pH is high, that's a base. If a pH is low, that's an acid. Okay, 39. Adding an acid tends to what to the hydrogen ion concentration of a solution?
Increase.
Increase, that's right. Acids increase the hydrogen ion concentration and drop the pH, which sounds kind of strange. You have to get used to how you talk about the pH scale. When something's low on the pH scale, it's an acid, and it's high in hydrogen ions. When something's high on the pH scale, it's a base, and it's low in hydrogen ions. So there's an inverse relationship on the pH scale because the pH scale is based on logarithms. But anyway, it takes algebra. We're not going to talk about algebra. 40. What is a buffer?
It resists change in the pH level.
Right. It's usually a chemical that's added to a solution that prevents pH changes. And anytime someone takes a medication or they receive an injection or they receive an IV, it will contain a buffer to prevent pH changes. Because if you change someone's pH by giving them a medication and they don't need their pH changed, then you've just caused them an extra problem. And so you want to you want to make sure that they have a stable pH and that pH is not changing if it's normal. 41. What is an organic molecule?
One that contains carbon.
Yeah, carbon and
Hydrogen.
hydrogen. It has to have both, and because there's one exception, which is CO2. Right, CO2 is a carbon compound that is not organic. What is an inorganic compound? So it does not contain carbon, and so we could say sodium chloride, which is NaCl. Let's see, that was all the ones that were listed in 24, question 24, all the salts, sodium chloride and hydrogen sulfate, magnesium sulfate, all of those are examples of inorganic. 43, what does it mean when it's said that water is a polar molecule?
One side has a positive charge and the other has a negative.
That's right. Okay, so polar molecules, the definition of polar I gave you to remember, and it'll become important later, is that opposite sides have opposite charges. So opposite sides have opposite charges. So the little hydrogens are positively charged in a water molecule, and the oxygen is negatively charged. 44, what is cohesive and adhesive? What is that? Cohesion and adhesion.
Cohesion is a molecule's attractiveness to itself, and then adhesion is attracted to an external surface.
Yeah, so cohesion, water we talked about is cohesive. Water molecules are like little magnets, so they like to stick to one another. This creates surface tension. And then water molecules also will bind to anything else that's polar, so they'll stick to other things that are polar. And lots of things are polar. One of the only things that's non-polar that you see often water come in contact with is fats, oils, or waxes. So if you freshly wax your car and water droplets fall on it, they'll turn into little beads and they'll just bounce off of the car because the wax is non-polar and water only bonds to other polar things. So 45, what takes place in a dehydration synthesis reaction?
Two monomers are combined with a water byproduct.
That's right. Okay, so and what's the whole purpose in dehydration synthesis? Now, of course, this requires an enzyme, so an enzyme controls it. But what's the whole purpose behind it? These two monomers that you're bonding together, it's impossible for them to bond together. They could never bond together unless an enzyme makes them what? What does the enzyme have to do to them to allow them to bond together? Makes them
Stable.
Unstable.
all right, makes them unstable. It makes them unstable so that they can bind together. So in dehydration synthesis, water is removed because the hydrogens and oxygen from the water actually were components of the original monomers. So when it's removed, they become unstable, and the monomers then have to bond together. And so the enzyme makes it to where the reaction has to happen, a building reaction. Synthesis reaction has to happen where before enzyme action, it would not happen at all. So what is hydrolysis?
Water added.
Yeah, water is added to split molecules apart. So really what we're saying is that hydrogens and oxygens are added to a molecule, added to a large molecule to break it down into smaller ones. Why is water added? Because the hydrogens and oxygens will fill in the missing pieces once the molecule is pulled in half. It will fill in the parts of the molecule that need to be stable. So hydrogen and oxygen play a role in organic reactions. Water plays a role in organic reactions. 46, which functional groups would cause a molecule like citric acid to be acidic?
Carboxylic acid.
Right, the carboxylic acid group, the carboxyl group, which is a carbon double bonded to an oxygen, single bonded to an oxygen and hydrogen. Sometimes it's written out as COOH. You'll see it written that way. Let's see. On a, like if you saw it written out in a question, it would be COOH. Sometimes you'll see that. That is just an abbreviation for the carboxylic acid group. If you just go on the web and look up any organic acid, like citric acid in the fruits that you might eat, and you look at the structural formula of citric acid, you'll see it contains a carboxylic acid group. We have to talk about those, and you say, why do we have to talk about that? We're not going to use that. I mean, maybe some people say that. You're going to see it because you have a huge chapter later on about acid-base balance. And these little carboxylic acid groups in amino acids actually help to keep pH balance in the body. In case you become basic, carboxylic acid groups donate a hydrogen, which helps to raise your pH. I mean, to lower the pH. And so these carboxylic acid groups are going to be important later on. Okay, 47. Which functional groups are present in amino acids? So we have two.
The amine group.
Yeah, the amine group and then the carboxylic acid group. Hence the name amino acid. Amino acid. Okay, let's see. So I have next, be able to recognize each of the functional groups if they were in a formula. So if you saw those in a question, you would be able to recognize the phosphate group or carboxylic acid group or amine group or hydroxyl group. You would be able to recognize those. We just did 4, didn't we? I think that's right. 49, cellulose is a... What's
Carbohydrate.
cellulose? It's a carbohydrate, that's right. It's a carbohydrate made up of many monosaccharides. Okay, 50. A long chain of simple sugars is called a
Polysaccharide.
polysaccharide, right? Polysaccharide. And a starch would be an example of a polysaccharide. A starch is made up of many glucose molecules, and of course that's why someone who has diabetes cannot eat a lot of bread, and because that bread in the body once they digest it, they'll break it down into just lots of glucose molecules, and those glucose enter into the bloodstream once it's absorbed and raise their blood sugar. So 51, animal starch that is stored in the liver and muscles is stored in the form of, anybody know? Glycogen. That's right. Glycogen is animal starch. So glycogen is important to us because anytime we eat extra glucose, we store it. If you go and exercise, if you go run long distance, something like that, then you replenish your carbohydrates afterwards. And what you're doing really is putting the glucose in the form of glycogen back into your muscles. So 52, the parts or words that denote a sugar. So what are parts of words that denote a sugar?
Glyco and oats.
So glyco, os, what's another one?
Saccharine.
Saccharine, right, is Latin for sugar. So any of those refer to sugar. 53, an organic compound with the chemical formula C11H22O11 is most likely a
Carbohydrate.
carbohydrate, right? Because carbohydrates consist of carbon, hydrogen, and oxygen with a 2-to-1 relationship between hydrogen and oxygen. Let's see, 54, an organic compound with the chemical formula C55H1O4 and O6 is most likely a
Lipid.
Lipid.
lipid, right? Lipids have lots of hydrogens, especially saturated fats have lots of hydrogen, which are the ones you don't want to eat, like bacon grease, lots of bacon grease. 55, what is the basic difference in the molecular structure between a saturated and unsaturated? Well, I just told you, really. It's all the hydrogens, but the reason that the saturated fats have more hydrogen is because they only have single bonds between their carbon chain and between their carbons in the chain, where unsaturated fats have double bonds, and the double bonds take the place of hydrogen. So unsaturated fats have less hydrogens. So this changes them chemically in the way they work, and the way that, especially the way that we see them and their physical properties. So saturated fats, an example of saturated fats would be something like bacon grease, lard, if you saw lard, or Crisco. So what would that be? That's solid at room temperature. Solid at room temperature. Where unsaturated fats would be what at room temperature?
Liquid.
Yeah, liquid.
Liquid.
They would be oils, like olive oil. Olive oil is one of the healthiest fats to eat. And then there's lots of other vegetable sources of oils that are good. So 56, the backbone to which fatty acids are joined in a triglyceride is a
Glycerol.
glycerol. That's right, a glycerol, which is a type of alcohol that we have in the body, and lots of it. All of our cell membranes have glycerol in them. Okay, 57. Why are proteins so important to human physiology?
Because we're made up of proteins.
So we're made of a lot of protein. And second, all of our
Chemical reactions are fueled by
chemical,
proteins.
that's right, all our chemical reactions are controlled by proteins. So we're made mostly of protein, but really there's kind of a debate. Some people think we might be kind of close to being equal in protein and fats in our body. We have a lot of fat. If you imagine, all our cell membranes are made out of phospholipids, which are fats. But filling those cell membranes are proteins. We have all these channel proteins and portal ways, and they're all enzymes. So kind of we're made up of a combination of fat and protein together, and so a lot of protein, but all our chemical reactions are controlled by proteins, which are enzymes. So what is a polypeptide?
A chain of amino acids.
Yeah, chain of amino acids, and just the smallest chain of amino acids. How many amino acids are in just small polypeptides? Anybody remember?
300?
Yeah, close to 300. And many polypeptides can be thousands, well over 1,000 or more amino acids long. So they get extremely complex. And of course, your DNA holds the instructions for building all of these proteins. 58, proteins have 4 levels of structural assembly. And so they're listed here. Let's see, the first says the blank structure describes a protein that is assembled from 2 or more separate poly chains. So these are out of order. Does anybody know what the first one is?
Quaternary.
So quaternary, that's right, quaternary, which is the most complex level of folding. This would be like you're building a machine and you have to use different parts. So different parts of the machine putting it together. Can everybody still hear me okay?
Yes.
Yes, sir.
Okay, it says my internet is slow, so I was just asking. Okay, so B says the blank structure of a protein describes the sequence of amino acids bonded in a polypeptide.
Primary.
Primary.
So that's the primary, that's right, primary. Then C says the blank structure of a protein includes additional 3-dimensional shaping that results from the interaction of the R groups.
Tertiary.
Tertiary.
Okay, that would be, that's right, tertiary. And then D says that the blank structure of a protein is a 3-dimensional shape that results from hydrogen bonding between amino acids, which is the first thing that kind of happens. And that would be secondary. So there's 2 different shapes that result when the hydrogen bonds start forming between these amino acids. You can have an alpha helix or a beta pleated sheet, so 2 different shapes that form. Okay, 59. What are globular proteins versus fibrous proteins? So these are 2 different shapes. Globular, probably if you think about it slang, you might think of the word glob, but that's not a science term. Globular comes from the word globe, and it means a spherical, so it's like a globe. And most globular proteins are enzymes or antibodies that would be in the body. So certain kinds of plasma proteins would be globular. And then fibrous proteins are like a fiber. They're long and thin, and most of those are structural proteins. Does anybody know what is the most abundant protein in your body? It's a fibrous protein.
Collagen.
Collagen, right? It's collagen is the most abundant in your body. And if you've ever seen a concrete foundation poured, you're going to get to this in your next chapters and when you study the skeletal system. That's why I'm mentioning it now. If you've ever seen a concrete foundation poured, before they pour the concrete, they put in iron bars. And these iron bars they call rebar. You've heard of reinforcement bar, rebar? Collagen is like the rebar, the protein rebar in your body that holds all your tissues together. So you have a lot of collagen in your body. Otherwise, your cells would fall apart and separate from one another without collagen. And that's what happens when you age, by the way. As you age, you get less collagen in your body. And what happens to you as you age since your collagen is depleted?
Your
Everything
skin gets
sags and
wrinkly.
wrinkles. Everything sags and wrinkles as you age because you lose your collagen. So it's like you're melting as you lose your collagen. Okay, so don't think about that. But anyway, 60. The suffix blank denotes an enzyme.
ACE.
ACE, right? Any word with ACE at the end is an enzyme. 61. The substance acted upon or changed by an enzyme is called the enzyme's
Substrate.
Substrate.
substrate. 62. What is a coenzyme? Anybody know?
Kind of just works with the enzyme to.
Yeah, that's right. It works with the enzyme in order to complete a reaction, and most coenzymes are
Vitamins.
Vitamins. So like vitamin B will be important in AMP2 when you talk about cellular respiration, because you have to have vitamin Bs to make ATP in the mitochondria. So that's why if you've ever looked on the ingredients in an energy drinks are full of vitamin B, because that's their scientific evidence that it's not just the caffeine, but really it is just the caffeine in an energy drink. But they fill them full of vitamins B6 and B12. 63, proteins may be blank by heat or acid.
Denatured.
Denatured, which means they lose their three-dimensional structure. And when they do that, they're destroyed. Their function depends on the
3D.
Yeah, they're 3D, they're three-dimensional shape. And blank are proteins that act as organic catalysts, and that would be enzymes.
Enzyme.
Enzymes. Very good. Okay, 64. What is the single most abundant protein in the body? And we've already said is
Collagen.
collagen. Collagen, right. 65, let's see, is that where we're at? Yeah. At 65, the monomers of nucleic acid molecules are called nucleotides, right? How is genetic material coded in DNA?
The nitrogen bases.
Yeah, it's the order of the nitrogen bases, which are found in the nucleotides. And we have A, T, C's, and G's, which is adenine, thymine, guanine, and cytosine, which we'll talk about more in just a minute. How does DNA differ from RNA? So DNA is?
Thymine is replaced with uracil.
Right. Yeah, DNA is double-stranded. That's right. And the sugars are different, right? They're actually named after their sugars. DNA contains
Deoxyribose.
deoxyribose, and ribonucleic acid contains ribose. Okay, so last chapter, the cells now, 68. What is spontaneous generation? What was that idea?
Cells arose from non-living matter.
Yeah, that cells just spontaneously arose from non-living matter. And scientists, of course, have tried to replicate that in a laboratory and never come close to that. And so spontaneous generation has been discounted and said, no, that couldn't happen. We've never observed that. And so it's kind of fallen to the side. And it goes further. They thought entire organisms came from spontaneous generation. And so later, the cell theory was developed by two scientists, Schleiden and Schwann, which were German scientists. And they said, no, cells only come from cells by cell reproduction. So the idea is life only comes from life. Life does not spontaneously generate. And so the cell theory says cells only come from cells. All the activities of the cells are controlled by the biochemical activities inside. And that the cell is the basic unit of life. Now, while I'm talking about this, what have you been hearing about the coronavirus this whole time? What do they say on the news all the time? What do people talk about? They say
Killing it, but it's not alive, so you can't kill it.
exactly. They talk about killing it, but it's not alive. It's only at the molecular level. So it's only made up of protein with some fat and a nucleic acid. So I think it's just layman's terms. You hear stuff like that all the time. Of course, some scientists want to say that viruses are alive, and so you can actually read about that sometimes and Discover Science magazine and stuff. They'll say viruses are alive, but it's a huge debate. Most people say no, most scientists say no, because ultimately, if you said viruses are alive, you would have to say, it would lead to saying that everything would be alive. And that would really be a problem in biology. I'd have to go back to school, have to take physics and everything. Let's see, 67. How does DNA differ from RNA? Oh, we did that one. Spontaneous. Okay, so 69. Blank is a sausage-shaped cell tapered on both ends and thick in the middle.
Fusiform.
Yeah, fusiform shape, good. So it's thick in the middle, like a kite string wrapped around a cardboard core. Okay, 70. Blank are thin, flat cells with angular outer edges.
Squamous.
Squamous, right? Squamous cells. So some people pronounce it squamous, squamous, or squamous. I usually say squamous. 71. Phospholipid molecules in a cell membrane are arranged with their, and this is a mistake that I have, with their blank on the interior. It should say what?
Hydrophobic
Right, hydrophobic tails.
cells.
So I made a mistake there on the review. It should say hydrophobic tails. And they're blank heads on the exterior, and that would be
Hydrolytic.
that's right, hydrophilic. Hydrophilic. And so phobia it means fearing, so water fearing tails, which are made out of fat. Remember, fat and water doesn't like one another, they oppose one another. So the tails face inward into the interior of the membrane itself. And then the hydrophilic heads, hydrophilic means water loving. And so philios is one of the Greek words for love. And so water loving. 72. What are the functions of proteins within the plasma membrane? So there's several of these. Proteins can act as,
To give it functionality.
yeah, that's what they do, but what are the different functions that proteins can give a membrane? So some protein, I'll give you a hint to begin with. So some are channel proteins, which means they're doorways through the cell membrane. So we have channel proteins. We have what else? And we're going to talk a lot about channel proteins, by the way, for the rest of the semester. After you do the integumentary system, there's going to be, and the skeletal system, then half of the semester is all about channel proteins and cells and how muscle contractions and nerve impulses and all that work. So you're going to talk a lot about those. So channel proteins, there's also carrier proteins. That's right, carrier proteins or channel proteins, but they need ATP. That's the difference from them. And then there are some that are cell identity proteins, right? Like the glycocalyx. The glycocalyx is an arrangement of carbohydrates and proteins on the surface of a cell that are used to identify cells in your body. So cell identity proteins. Then enzymes, and there's one more, which is motor proteins. Motor proteins make parts of the cell move, like cilia or a flagellum or something like that to make a part of a cell move. Okay, 73, define and understand hypertonic, hypotonic, and isotonic solutions. So when you think about these three terms, some students get them confused. You're going to be talking about them a lot if you're going into nursing later on. You talk about all these solutions because your body is made up of these different kinds of concentrated solutions. What you're referring to is tonicity, which is studying osmotic pressure in the body. And that deals with the concentration of dissolved material in body fluids. And so what does the word hyper mean? The Greek word hyper
Increased.
High.
high, right? And think about tonic referring to salt or electrolytes. And even though it doesn't mean that, it refers to solution, but kind of think about it meaning salt. So hypertonic is a solution with high salt. Hypotonic would be what kind of level of salt or dissolved material?
Low.
Low salt. And then iso means equal in concentration of dissolved material. So one of the keys that you need to remember forever is remember that water always flows toward a hypertonic solution. Water is attracted to a hypertonic solution, and we call that in AMP2, we'll get into that a lot more, it's called osmotic pressure, that water is drawn toward a hypertonic solution. So that's going to tell you the direction of water. Water moves toward the highest amount of salt. So in a hypertonic solution, if you place a cell in a hypertonic solution, That means that the solution on the outside of the cell is higher in salts than the inside of the cell, and water is going to leave the cell and dehydrate it. That cell is just going to shrivel up, and that's called crenation. Crenation. And then hypotonic solution means low salt. What if you put a cell in a solution that's low in salt? That's just like pure water. What's the cell going to do? It's going to swell and it could explode, which we call cytolysis. It could explode. And then isotonic is just right. It's like the Goldilocks, just right. It's just right for health and normal survival. And when you give someone an IV solution, it will be isotonic to their body fluid, isotonic to their blood. It's going to be equal because you don't want to change anything. You just want to give them what's right, just what they need to survive. So IV solution would have just the right amount, just the same amount of water, just the same amount of salts, glucose, same pH as normal human blood would. That's going to be normal. That's going to be important to you that you understand that. These solutions, I think I gave you in one of the lectures that the concentration of body fluids is is measured in a unit called milliosmoles. And you'll learn in A&P2 that our body concentration is around 300 milliosmoles. And when you start memorizing IVs, if you're going into nursing, you'll memorize that a normal IV solution is around 270 milliosmoles, and that would be isotonic to body fluid. That would be the standard solution that you would give someone. And there's a lot of other different kinds of ringer solutions that you give before surgery and other things. So you have to memorize those things later on. You get more into it in A&P2. This is just an introduction. Okay, 74. In osmosis, water always moves toward and so here's what I just told you.
Hypertonic.
Yeah, hypertonic solution. 75, you open a bottle of perfume in a bedroom, and soon it fills the entire room and the bottle's empty. That would be an example of what?
Diffusion.
Diffusion, right, diffusion. 76, active transport requires
Energy.
energy, right, in the form of what?
ATP.
ATP, right, ATP energy. Okay, 77, the process of a cell engulfing a large particle and bringing it into itself via a vesicle is called?
Endocytosis.
Okay, endocytosis, and specifically since it says a particle, what kind of endocytosis?
Phagocytosis.
Right, phagocytosis, which means eating. Phagos in Greek means to eat. Pino means to drink, and so pinocytosis is the cell taking on fluid. 78, many gland cells release their secretions by means of?
Exocytosis.
Exocytosis, good, right. 79, what might happen to material that is phagocytized and taken into a vesicle once inside of a cell? So it could be digested by lysosomes. So it could be digested. And this would happen if it was like a macrophage, which is a white blood cell that's the big eater, and it engulfs, and then it digests whatever it engulfs. When you get sick with a bacterial infection, why are the bacteria making you sick? What are they doing to you?
Attacking the cells.
Yeah, what are they doing when they attack the cells? The whole purpose in them doing that is because they're eating you, right? When you get sick from bacteria, they're eating you. They're alive. They need food. They're eating you. And the way your immune system gets rid of the bacteria is the cells, your white blood cells, eat them first. And so isn't that weird? That's what goes on at the cellular level. It's like this giant battle going on all the time at the microscopic level. Okay, let's see. It gets me excited. I like to talk about it. Let's see. What is the next one here? So 80. A cell having a resting membrane potential has what characteristics?
An electrical charge.
Yeah, it's electrically charged. The outside of the cell is what charge? Is it positive or negative?
Positive.
Positive. And the inside of your cells are always
Negative.
negative. And what kind of pump creates that scenario where you have positively charged on the outside and negatively charged on the inside? What kind of pump maintains the electrical potential of your cells?
Sodium, potassium.
Sodium potassium pumps, right? So remember those sodium potassium pumps. I just have been talking about them in AMP2. You start off in AMP2 with how the heart works, actually talk about endocrine system and then blood, and then you get to the heart and you come right back to sodium potassium pumps. There will not be sodium potassium pumps in the endocrine system. I'm sorry, in the integumentary system, which you do next. And there will not be sodium potassium pumps in the skeletal system, which is after the integumentary. But then when you get into muscles and nervous system, it's constant. They're always working 24/7. And you have to remember they're working in the background, sodium potassium pumps, to maintain the potential of cells. Okay, 81. Which RNA is responsible for bringing the amino acids to the ribosomes during protein synthesis? That would be the
Is that mRNA?
Transfer
so
RNA.
yeah, transfer. That's right, transfer RNA, tRNA, tRNA. It transfers. It's an escort molecule. So if you haven't watched the videos, I think I have 2 videos on photosynthesis. Not photosynthesis, protein synthesis. I made one, and then I gave you a tutorial of one of another video. Be sure to watch those if you haven't. What is the role of mRNA, transfer RNA, and rRNA? So mRNA is messenger RNA. What does it do? Ultimately, it carries the message of DNA out of the nucleus and brings it to the ribosome.
The Robert.
And ribosomes are made out of protein and rRNA. And so that's where rRNA comes in, because ribosomes are made out of protein and rRNA, which is ribosomal RNA. And then tRNA, what does tRNA do? It brings the amino acids according to the mRNA's instructions. It brings them and drops them off. So again, I know you've just been studying this today and yesterday, and today's your quiz. It's due today on the cell. Make sure you watch the videos on protein synthesis. There will be some questions on them, on the test on it. So I try to give you a simplified version. When I used to teach high school, and when I taught high school, I'm just giving you that simplified version, high school version. So you should be able to do it easy. 83, complete the transcription and translation of the DNA template. Okay, so if anybody, let's see, is anybody writing on the screen? And so if you want to fill in, we'll just fill in a couple of these. I'm going to have to enlarge it. You can't enlarge the review if you can't see it. There's a little magnifying glass in the top left corner, and if you click on the zoom in, then you can magnify it. So I don't know if you see it magnified as I do right now. Does anybody magnify? Or is it still tiny? Is it small or is it enlarged?
I had to manually enlarge mine.
Okay,
Mine's
so
enlarged.
that's what I did. I've enlarged, and so T, if you looked at the first one here, a thymine will bond with an adenine. Okay, so adenine is brought to the thymine by an enzyme called RNA polymerase. So RNA polymerase will bring RNA nucleotides to the DNA template strand. So again, watch the video I made on it. I make it real simple when you watch it. And so this enzyme, RNA polymerase, brings to an A, it brings a U because there's no thymine. So it brings a U to cytosine, it brings a guanine to a guanine, a cytosine. And so what we have is a little pairing rule in DNA. This is called the DNA to RNA base pair rule. So in DNA, if we have adenine, thymine, guanine, cytosine. What would that pair with in an RNA molecule? Well, A's would bond. If A was in DNA, a uracil would bond with it. Now, with thymine, it would be adenine because we still have adenine. Guanine, cytosine, cytosine to guanine. Okay, so if we're down to cytosine here in the DNA, what would bond over here in the RNA? It would be a guanine. Okay, and then to guanine, we would have a cytosine, and then cytosine to guanine, and guanine to cytosine, so all the way down. And then at the end, I'll just, let's see, do it. We have a guanine, and then U, and then A at the bottom. Okay, so when you fill that out, there is a page, I think it's page in your textbook. I don't have the textbook to where I can grab it, but I think it's page 108 or 107 in your textbook that has this illustrated for you. And I think I tell you about it in the video for you to look at. If we look at the codons are the coding words. They're every 3 units that are on the mRNA. And then tRNA is represented here by 3 blanks and 3 blanks under the anticodon region and an amino acid. So if we were to bind RNA to RNA, this is real easy. This is the RNA to RNA base pairing rule. And so if we have RNA to RNA, adenine bonds with uracil, uracil bonds with adenine, cytosine bonds with, or this is guanine, bonds with cytosine, and cytosine with guanine. And so A's bond with U's, U's bond with A's, and so you see how we're bonding over here. And then guanines bond with cytosines. So our very first anticodon that bonds with AUG is UAC. The next thing you would do in order to find the amino acids, you would have to look on the codon chart that's in your book. And I don't have my book, so I can't tell you what page number it's on. But what you always read when you're looking at the codon chart is you always read the codon strand, which is right here on the mRNA. That's what you always read to find out what the amino acid sequence is. And so the very first amino acid that's in the sequence is the start. It's the start codon, which is methionine. Methionine. Methionine is one of the 20 amino acids. So it would be lined up. And then I don't have my codon chart to be able to look it up to tell you what the next 2 are. So you would have to look it up in the sequence. And again, what do you read to find the amino acids? You look at the mRNA sequence. The last, though, if you have your codon chart, did anybody find the codon chart in your book? What page it's on? Do you have your book? Let me, hold on just a minute. I'll be right back. I'll get my book and tell you what page. Okay, in the textbook. So let's see, page 108. So page 108 is where this is summarized, but your codon chart that's in the book is on page 103. And so I'll go ahead and tell you what these are now that I have my book. And so if you look at CGC, which is the sequence here, CGC, then on the codon chart on page 103, CGC, anybody find it first? CGC is arginine, R-A-R-G, arginine. Okay, and then you look at the next 3, which is GCG and GCG. Let's see, on the codon chart, GCG is alanine. That's right. That's right. Very good. Alanine. And then the last one is UAA. And the last 3 on every strand is the stop sequence. So stop. And so there's 3 stop sequences. And what you learn when you look at the protein synthesis is you learn that DNA is organized a lot like computer programs. Our cells have a programming language in them for all the proteins in our body. And that's amazing. It's so much like a computer program that someone like Bill Gates and Microsoft, they're working on designing a vaccine for the coronavirus because they know about DNA and programming, and they're programming biological molecules to be formed. So it's pretty amazing of how much our DNA is like computer programs. If you've ever written a computer program, I started out as a computer science minor, but I got bored sitting behind a computer so long typing algorithms. But when you type in lines of code, you always have start sequences and ending sequences, stop sequences. It's just like it's a language. So even our DNA has punctuation. It has start and stop in it, which is amazing. That's amazing. I mean, you find a language in living things. That's amazing. Okay, 84. I'll move on. I'll get on a soapbox and start talking. We're almost through. 84, during cell division, when does DNA replication take place? So DNA replication takes place during interphase, and if you remember the S phase of interphase, the S phase. And then you have true or false statements. A says the nucleotide sequence in an mRNA codon is the exact copy of the DNA triplet. Is that true? And is this mRNA here the same as the DNA here? Well, it says the same message, but it's not the same, right? DNA starts off with T and the mRNA starts off with A, so they're not the same. So A would be false. B says the nucleotide sequence in an mRNA codon is the exact copy of the DNA triplet that coded for it, except that uracil is substituted for thymine. Well, that's still false. In other words, if we just put U's for T's, it still wouldn't say the same thing. So B is false. C says the nucleotide sequence in tRNA, which is the anticodon, is the exact copy of DNA triplet. Well, that's not true because DNA contains thymine and RNAs contain uracil instead. So that wouldn't be the same. And you can see here that UAC is not the same as TAC, but it's getting close. D says the nucleotide sequence in the tRNA, is that where we're at, T? Yeah, let's see, nucleotide, yeah. D says the nucleotide sequence in the tRNA anticodon is the exact copy of the DNA triplet that it coded for, except that uracil is substituted for thymine, and that would be true. D would be true. If we took the DNA strand, which is TAC, and we just substituted U's for T's, then we would end up with the anticodon sequence. And so you can see if we had UAC, this is TAC, and we went all the way down the last two, instead of being ATT, which is in DNA, it would say AUU, which again would pair with the codon UAA. So again, if you haven't studied protein synthesis, watch those videos and it'll be easy to you. If what I'm talking about now is complicated, it'll be easy once you watch the videos. E says the translation converts DNA message to RNA, while transcription writes the RNA's message into a protein, and that's the opposite. It should say transcription converts DNA into an RNA, and then translation writes the RNA's message into a protein. So we're almost through now. 86, what is a gene? Define gene. So I'm asking you, anybody still there?
Yes. The section of the chromosomes that has the DNA.
Okay, so it's a section of DNA, that's true. A section of DNA, but what does it do? You inherit your parents' genes. What does it do?
Produces proteins that has the chromatin.
That's right. So it's a gene is a recipe for a protein. So a gene makes the instructions for making a protein. It's the recipe for a protein that you have in your DNA. And then 87, if tRNA had UGG anticodon, it would attach to which mRNA codon?
ACC?
ACC.
ACC, right? ACC would be the codon. And then what's a point mutation? What's a point mutation?
Error in a gene.
Yeah, so I'm going to go back and let me clear my screen now. So if you look at this DNA template sequence that's here, a point mutation would be suddenly changing, say, this A that's second. What if we changed it to a U? Okay, so if we change suddenly one nucleotide to something else, what would that do? Would it be the same
No.
code? No. And so, in fact, it may totally destroy the gene itself if we have no start. And so TAC is the start at the beginning of the DNA sequence. If we just changed one nucleotide, one point in the gene, then it entirely can change the message. Now, sometimes it doesn't, but it could potentially completely damage the gene itself and therefore cause a disease, which we think about as a mutation, which like hemophilia would be an example of it, and other kinds of disorders could be a mutation that occurs in someone. Okay, anybody have questions about the review? Any last minute questions? We've spent about the same length as a class time, about an hour and 20 minutes as class time. Anybody have any questions?
No, sir.
No, sir.
Okay, well,
No.
study and do well. And don't forget about your quiz that you'll be taking tonight over cells, and study protein synthesis, because it seemed like everyone knew things except for the details of protein synthesis. Okay, then, if you have any questions, be sure to email me. Everybody have a good day.
Thank you.
Thank you.
Bye-bye.